Hello. The next chapter in here, chapter 5, is going to be DNA tools. And with that, so these are going to be different ways in order to be able to manipulate your DNA. So with that, so your gene of interest. So with that, so the first step is what are called restriction endonucleases, or just simply restriction enzymes. So with that, so they cleave very, very particular strands of or sequences of DNA. So with that, so usually the cleavage site is going to be palindromic. So as you can see, in this particular one, it'd be CCGCGG. Right, but it's the same going this way for the complementary strand. CCGCGG. Stuff like that. So that's what I mean by a palindromic. There's a bazillion of these things out there, something like that. And so, but there's and so they can target a lot of different sequences. So like that. So after they cut, they don't necessarily have to just chop like this. They can actually leave what are called sticky ends. There certainly are a bunch of them. EcoR5 is the classic one that leaves what's called a blunt end, or just, the DNA stops. If you have a sticky end, you've got a short stretch of single-stranded DNA that's left over, and stuff like that. And so this would be complementary. You can use that to be complementary to a new piece of DNA. We'll talk about that in a second. So you can either leave it where the bottom strand is sticking out, or you can leave it where the top strand is leaving out. So or three, you can sort of 5' protruding, 3' protruding, or blunts with that. So you can use that to be able to put DNA together. So let's say you have this blue and this blue gene and pink gene here. So by that, so if you cut them both with EcoR1, which is a pretty classic restriction enzymes, they're going to leave these ends here. So with that, so along with this overhang, so you can take this overhang and have it be complementary over here. So and it'll stitch it together with the DNA ligase. So with that, or you can take this end and have it extend over here. But that's how and ligate them together. So it's a pretty slick way of putting this DNA together. We do this all the time in my lab. It works really well. So the other thing we can do is what's called electrophoresis, and so this is a nice analytical tool to be able to separate your materials on the basis of size. And usually you stick it at one end, you stick your the mixture at one end and let it flow. And so these are molecular markers, stuff like that. So for protein gel, for proteins, you typically use an acrylamide gel. So this is your classic sort of SDS page. If you use that, the SDS just means soap. The page is polyacrylamide gel electrophoresis. What's going to happen is it's going to denature all your proteins, so unfold them. And so using the SDS, it puts a charge on there, and so generally things move relative to how large they are. And the smaller it is, the faster it's going to move. And so right, so you think about running through a playground, right? A little kid is going to be able to run all over everywhere, but their parents, they're going to be taller, right? It's going to take them a lot longer to get through all that equipment, stuff like that. So you can separate out things by size, and so you can do this with both DNA, where you're looking at the base pairs, or you can look at the actual molecular weight here, something like that. So now once you have them separated, so that provides quite a bit of a clue as to what it is. But if you want a probe to figure out, where is my particular gene or my particular protein, whatever, stuff like that. So you can actually do what's called blotting with that. So you can take whatever the gel, either the agarose gel or the acrylamide gel, afterwards, and you can transfer that. Put a charge on this and transfer the material to a nitrocellulose sheet. So you just literally stick the sheet on there, apply a charge, and the materials will actually move onto that onto the sheet. You can then take the sheet, and then you can do a probe. So if this was DNA, stuff like that, so you could have a DNA probe that's radio labeled with P32. That was the original way you can do it. You can also do it with fluorescence or things like that, stuff like that. So this probe, you know exactly what the sequence is. It's going to find its complement, and in this case, it's here. So with autoradiography, you can use the the radioactivity of the P32, put a thing of X-ray film on it, and it can show you, oh, my stuff is this particular, of all of these sorts of things, mine is this one right here, the one that I'm interested in, because it is complementary to this. So, so this technique was originally done with DNA, by Edward Southern, stuff like that. So it's called a Southern blot, stuff like that. So, but then they realized, hey, you can do this with RNA. So a group of, I believe, is out in Stanford, developed this, and they thought it'd be hilarious because this is a Southern blot, the complementary, right? Because it's RNA, to be called a Northern blot, stuff like that. So and it's just sort of a shout out to Southern, stuff like that. But it works the same way. Oh, and then proteins, because now we're talking about directions. They decided, hey, the term got coined Western blot. So if you hear about Westerns, in the molecular biology sense, you're going to be doing this probing, but it all works the same way, right? You separate out your sample, the mixture, by size. You transfer it to a membrane. You then can come in and then you stain it. So with DNA, you can probe it with complementary with DNA. You could also do it with RNA. Same thing with this, RNA probes, DNA probes, things like that. With proteins, you typically use antibodies that will bind very specifically to a particular protein, and then you can detect detect the presence of that antibody. Wherever that antibody goes, that's where your stuff is going to be, things like that. And so these things are very, very powerful, very sensitive, and it works really well. So it can. Okay, so with DNA, right? How do we know we've got the sequence that we do, right? How do we know we've got the A's, the G's, C's, and T's in the correct order? One of the first practical ways of doing this was developed by Frederick Sanger, and so it's still used. We use this all the time. I'm going to be dropping off some samples later today, and it works. It's a very robust way to do this. This is how they originally did the Human Genome Project. So no problem. The way this works is that it uses a DNA polymerase in a PCR type reaction, which we'll talk about in a second. But what it does is it puts in the normal DATPs and that sort of stuff, the monomer nucleotides. But it puts in a small amount of the dideoxy. So it has a it removes the OH at both the 2' and the 3' part of the sugar. So like that, there's also it's also fluorescently labeled. Okay, and so what happens is the polymerase will come in and start extending out the chain. Okay, and so like that, and so if it accidentally adds in this one here, the chain is gonna stop because there's no 3' hydroxyl to be able to move that chain along, so it stops. So you're going to have a fragment that's this long, okay? And then, but on a different chain, you're going to have one, and the polymerase is going to chug it along, and then it accidentally grabs this one instead of that one. So now you got a fragment that ends in this one, and so on and so forth. So you're going to have a huge mixture of different chain lengths, okay? And they're all going to be at the end is always going to be have a different fluorescent dye. Stuff like that, corresponding to each individual nucleotide, excuse me. So that, and so what you do is you put this mixture on what's called a capillary electrophoresis. It separates things out by size, and stuff like that. And so what you can do is you can actually literally read what's coming off of here, because you get what happened here? You can then read the order of how the different colors that are coming off, and that's going to correspond to, T, A, G, T, things like that. And so you can read that coming down the pipe. So for that, it works really, really well. So for that, or at least it can. So for that, there is what's called next generation sequencing, and this is used. I don't know. I'm not as familiar with this. The techniques. There's several ways that you can do it, but in general, what happens with this is that you assemble a large number of sort of 20 to 25 base pair fragments of the DNA, stuff like that. So you do a whole bunch at the same time, and you can figure out what these sequences are. So you can get a million reads at one time, and then you find, and then you use computers to be able to assemble it all and stuff like that. And so like all of the different fragments should come together. There should only be one way to be able to put all these DNA fragments together to get your gene. And so this is the way that you're able to get genome sequenced so inexpensively. Stuff like that. It's really amazingly powerful, especially when you combine it with the computational power that we have now. So so this is getting increasingly important. So the DNA itself is actually very easy to synthesize, stuff like that. So it's actually pretty much completely automated, and they actually build the DNA on these solid supports, and so they add it one nucleotide at a time. And so what happens is you've got a resin bead, so it looks like sand, things like that, and you start to add it one base pair at a time. And so you couple the reaction, and then you add on your base pair, and then you can just wash, right? Your resin is going to be stuck with your stuff. Everything else is going to get washed away, and then you can go through the cycle again and add another wash, things like that. And so you can start building these up. Now it's got the quirk that it actually builds up 3' to 5', but it doesn't really. That's sort of on that end of the thing. When it comes out, it'll be just what you need, stuff like that. So it's really simple. You literally just get online. This is the place that we use. They literally just down the road, stuff like that. So you can just fill in what you need, your name, sequence, how much you need other sort of stuff, and you hit go. And usually by the next, if you get it done before the end of business, stuff like that, it'll be, they'll drive it over, the next day, stuff like that. It's amazing, and it's not that expensive, maybe like 10 to 15 cents per base pair. So if you need a 25 base pair thing, it's going to be $2 to $3, stuff like that. So it works. It's amazing how efficient it's become. So one of the things that you can do with these primers is go through what's called the polymerase chain reaction. And so this was another one of these Nobel Prize winning inventions. The sort of the mythology is that Kary Mullis, it was sort of a eureka thing, but it turns out like all stories, it's much more complicated than that, and he got a lot of help. He was working at Cetus Corporation, which was one of the very first biotech companies out in San Francisco, and they and sort of like his colleagues ended up getting putting a lot of work into this, stuff like that. So, but it allows you to amplify a very specific region of DNA. So you can literally start with one molecule of DNA, and if you know what the sequence is, or if you sort of have an idea for what the sequence is going to be, you can make large amounts of that section. Okay, and all it is it's and what you do is you have a primer that's complementary with. I'll show you in a picture in a minute, to the 5' and 3' end of your gene that you want to amplify, something like that. And then you go through these heating cycles. And originally, so you have to go through this annealing or this melting step. So you melt the DNA up to like 95 degrees, and then you go through an annealing step. Now so now you've got single-stranded DNA. When this cools, the primers actually anneal before the full DNA comes back on there, and so you're going to get the primers annealing, and then you go through a chain extension step where it extends out from those primers. Now, originally when this was done, there was literally a robot that took your you had three different wells at different temperature, and the little robot that would move it between the two, and then stuff like that. And the problem is, once you heated it up, stuff like that, so it would destroy your DNA polymerase, stuff like that. And so after each step, you then had to add more polymerase and then go back through, stuff like that. So, but one of the amazing things about the way the Kary Mullis developed method is it used this thermostable DNA polymerase from Thermus aquaticus, which is from these hydrothermal vents at the bottom of the ocean, stuff like that. And so these are organisms that grow at 100, 110 degrees Celsius, and stuff like that, so they can stand these sort of extended times at high temperature. And so the chain extension steps actually usually like 72, stuff like that. And so all you have to do is mix everything together, and now they've got machines that'll do this cycle, this heating cycle automatically. So you literally just hit go, you program it, you put your tube in, hit go, and then you come back, an hour or 2 hours later, and there you go. Your stuff is amplified. And so the way this works is that, so if this is your target gene, you're going to want sequences that's complementary, that's for this part of the gene and then this part of the gene. Now this one's going to encode for that top strand, this one's going to encode for the bottom strand, and so you need the reverse complement for this, because remember it's going to get sent, you need to go 5' to 3' on this one, so you have to be careful. But as you go through this heating step, it's going to extend out this chain, stuff like that, in both ways. And so again, it's that sort of semi-conservative method. And so when it goes through another cycle, another set of primers is going to bind here, 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 and here, and again they're going to go out here. Now if you had this blue strand and this primer here annealed to it, right, so you're going to get that short strand, and eventually as you go through this, this is going to be a very small amount, and you're going to make tons and tons and tons of this sort of shortened strand DNA, stuff like that. So the amount that this gets amplified is 2 to the number of cycles that you have. Typically it's like anywhere between 25 and 50 cycles, usually 30. We usually use 30, stuff like that. So so you're going to make a lot. You can make a lot of PCR fragment pretty quickly. So and what you can do with that for, if you want to be able to do this, is to be able to take out a gene from a genome, things like that, and then insert that into an organism to make your protein recombinantly. So the classic case is something like insulin. Okay, and so you take out the DNA from the pancreas. Now you don't isolate the DNA, you isolate the RNA, right? Because you need all those introns removed, all those inserted introns removed, stuff like that. So you use this reverse transcriptase to take this, and actually what you do is you say, okay, I've got this poly A tail, so what you can do is you can you put in a poly T, and it'll bind here, and then you can make that into DNA. Once you've got that first strand of DNA, then you can make your double-stranded DNA. You can then join that into a plasmid, and then put this into an E. coli, and then the E. coli you can use that to be able to the transcription translation machinery inside the E. coli to make a lot of protein. And so they still use this today. They still have E. coli that makes this, and it's can be incredibly cheap, particularly at scale, things like that. So now this here is pretty much becoming less and less likely, like that. And so because you can actually do the DNA synthesis of it's got so inexpensive and easy to do that you can actually just get a gene synthesized just from scratch, stuff like that. So I've literally ordered genes from pathogens, so like plague and things like that, just as individual gene. I mean, it wasn't anything infectious, but you can do that. Stuff like that, and you don't have to you don't have to handle the organisms at all. So this is really great it's also great if you're using, let's say, animal sources, right? You don't have to worry about any sort of animal contamination or viruses or anything else like that. You can just order the gene, and you literally go on their website. So this company I use. They're great. Atom, stuff like that. So and you go through this step, and it's you can just say I know what the amino acid is, so let's build a DNA from that. You can also just take your DNA and optimize it, because what you can do is you can say, I want this to be expressed in E. coli. So different organisms use the codon. The codon genetic code is generally the same, but how the tRNAs are utilized. Some prefer some codons, some prefer the others. So you can use that, and they'll take that into account. So if you have a human gene and you want to make it an E. coli, you can give it the codon patterns for E. coli. You can get in there and say, okay, well, I don't want these restriction sites in there. You can add cloning sites, whatever. Optimization, and then you can get it put into have it put into a plasmid that you want, stuff like that, and then how much do you want, and then literally you hit go, and then like 2 weeks later you get a little FedEx envelope and it's got your gene in it already in a plasmid ready to go. It's amazing, and and it's actually not that expensive, and it's gotten cheaper and cheaper over the years. So this is really cool technology, and it can really speed it up, stuff like that. So okay, thank you.